So for extra credit, I did draw transformation, transduction, and conjugation. So for transformation, I actually drew two different strains of the same bacteria. So I drew a viral strain, non-viral strain. The non-viral typically doesn't cause disease, but the viral strain does. So for any bacteria, once it dies, it releases its DNA into the environment. And once the DNA is in the environment, any similar bacteria that is able to bring its DNA into its cell can. And it kind of integrates the genes. So it can have some of those genes in it from a different strain. But in this particular case, the viral strain or viral bacteria die. So when the viral bacteria die, they do release the DNA into the environment. And once the DNA is released to the environment, sometimes there will be a nearby cell, i.e., the non-viral bacteria. And a non-viral bacteria is able to actually bring in the DNA from the viral bacteria into its own cell. And it uses some of its genes. So if the genes are integrated into the non-viral DNA that allow this bacteria to become pathogenic, if they join together, this bacteria can now become pathogenic because now it holds those genes that enable it to have those different functions or those virulence factors that enable it to become harmful. So then it then can transform into the viral or the pathogenic strain. So for transduction, we have a bacteriophage and a host cell. The bacteriophage attaches itself to the bacteria or the host cell and it injects its DNA into the host cell. So some enzymes actually break down the host DNA. And once the bacteriophage's DNA is kind of in there and the host DNA is breaking out into fragments, it is able to replicate and make more bacteriophages within the cell. So once all the DNA is kind of put into the bacteriophages, you can see that some of the bacteriophages actually hold the DNA of the host. That's because the replication of bacteriophages or any viruses typically aren't perfect. So they do take a little bit DNA of the host cell and kind of bring it into their own DNA. It's usually by accident because I'm pretty sure they can't do it on purpose because viruses are not alive. But once the bacteriophages kind of like are formed, they can burst out of the cell, causing cell lysis. So lysis is just cell death. That's what happens in the lytic cycle in particular. I'll explain after. But the bacteriophage, and I chose to use a bacteriophage with a host cell to show how this works. The bacteriophage then, kind of like at the start, it attaches to a recipient cell and injects the DNA into the recipient cell. Then the DNA of the recipient cell actually takes in the bacteriophage's DNA. So the recipient has a little bit of host cell DNA, bacteriophage DNA, and its own DNA within its genetic code. So any other bacteriophage that does combine to it, when it does have the same processes of breaking down the DNA, it'll break down DNA that has host cell DNA, recipient cell DNA, and its own bacteriophage DNA. So it kind of just multiplies from there. So it can pick up like different genes and stuff. So this cycle that I drew is the lytic cycle. The lytic cycle does end in cell death because the bacteriophages burst out of the cell, and cells can't survive if the plasma membrane has a hole in it. Can't really control what comes in or comes out. There is the lysogenic cycle, The lysogenic cycle does have a bacteriophage that connects and attaches to the host cell. Bacteriophage then injects its DNA, but instead of it going through like the cell lysis like it does here, it kind of remains like this recipient host cell where the bacteriophage's DNA actually stays in the host cell's DNA. So the host cell DNA will actually just replicate with the bacteriophage's DNA in it. So any viruses that kind of like once they get into you, like herpes, this is kind of how they function. And when the cell replicates, it just remains as is. So they don't cause your cells to die like they do in the lytic cycle. So for conjugation typically occurs when bacteria that have like some type of factor or plasmid kind of transfer that to another bacteria that does not have it or lacks that factor. So there are different kinds of factors. There's the F plasmid, fertility plasmid, that's what it stands for, which just enables it to grow a pilus. They have the R plasmid, the resistance plasmid, which makes it resistant to some antibiotics. And they do have a virulence plasmid, which helps it kill any other bacteria around it to kind of eliminate like competition basically. So for this example, I use the F plasmid. So the F plasmid, you do have a cell that does have the plasmid. So once it senses a similar cell nearby that doesn't have a plasmid, it sends out or extends a little protein-like structure called a pilus, and a pilus attaches to this cell and pulls it in and brings it close. So once the cell is close, the plasmid is able to send one strand of the F factor or the F plasmid into the cell that lacks that. So it's kind of like DNA replication, because in order to make a complementary strand of DNA, you only need one parent strand. You don't need both the parent strands to make one complementary strand of DNA. So this F plasmid in the cell gives the cell that lacks it one single strand, because it can make a complementary strand and form an actual F plasmid as seen here. So once the F positive cell gives its single strand over, it's just able to kind of form the other half of the complementary strand, kind of like replace it. So then you end up with cells that are both F positive, but for this cell, now these cells can go and transfer the genes as well to other cells that don't have the same plasmid as it does.